Hello, everyone, and welcome to our 14th lecture in our course on brain and behavior. And today we're going to cover research methods. So this is a single lecture unit, and we've been talking throughout the course already about the things that we know about the brain, about brain structure, brain activity. We've seen pictures of the brain. We've seen graphs of neural activity, action potentials. But we've never really talked about where those come from and how those techniques work. So now that we've seen some of the techniques' results, we'll talk about the techniques themselves and how we acquire data about the brain in a variety of ways. Okay, so we're talking about research methods, and we'll talk about a couple of experimental designs. And these are two sort of big categories that we mentioned at the very beginning of the course, but haven't gone much into depth in terms of the rationale behind those designs. So we'll do that today. And then we'll talk about categories of technique. And one of the big categories is imaging methods. So taking pictures of the brain. Now, those could be pictures of brain structure, the parts of the brain, or those could be pictures of brain activity, what we call functional images. So those are two different concepts, how the brain is built and what the brain is doing at any given moment. And we'll look at other ways of measuring actual individual action potentials, and as well as techniques for measuring groups of action potentials, electrical activity in general. And then we'll see that in terms of brain activity, we can measure it, but we also have some relatively new ways of modifying it, of activating part of the brain or shutting part of the brain down and seeing what happens. So what we'll do is see that there are different ways to approach this question. What is the brain doing? How is the brain built? And again, we'll talk about two broad categories of experiment here very soon. And we'll see the effects of neural activity. And what do I mean by that? I'm not talking about a psychological phenomenon. We've been talking about that for a while. What I mean here is that we talk about action potentials, neural activity, but that neural activity has a number of consequences in a physical sense. It obviously impacts electrical activity, and we can detect that with the difference in charge across a membrane or across regions. There are chemical signals that a neuron releases, neurotransmitters. There are also magnetic fields that the brain generates. So neural activity is not just a signal that gets sent from one neuron to the next. It's a number of different kinds of signals that we can detect and measure. And as we talk about these techniques, we'll talk about pros and cons. So no technique can do everything. Techniques that tend to measure at the small scale aren't very good at the large scale. Techniques that are very fast tend not to be very precise in terms of space, and vice versa. Techniques that can measure a broad area tend not to be very fast or very precise. So we'll see those various pros and cons. We'll see why you might choose one technique over another. And then we'll see some applications, not just in terms of basic science, our understanding of the brain, but also some clinical applications. How can we use these techniques to help someone with a psychological disorder, for example? So those two big categories, how do we know what we know? Well, we have terms like somatic intervention, somatic variable, behavioral intervention that we see here. And the question is, what do these terms mean? Well, broadly, the word somatic means having to do with the body. And really here, somatic for us is going to mean the brain. We care about the brain in this course. The brain is the seat of our psychological processes and experiences. So we're going to see here in a moment that we can either change something about the brain and see what that does to behavior and psychological experience, or we can change something about behavior and see what that does to the brain. So we'll start with what's called the somatic intervention approach. So here we manipulate the brain in some way. Historically, this is often done by changing brain structure. Perhaps a part of the brain is removed. Now, ethically, of course, we cannot go in and remove part of the brain on purpose just to see what happens, but part of the brain might be removed for surgery. to relieve the symptoms of a medical condition like epilepsy. Or part of the brain might be removed accidentally by a traumatic experience, by, for example, lots of our knowledge of the brain and comes from lesions due to injuries from war. So whether the injury was sort of intentional or not, the brain can be changed structurally, and then we can see what that does to cognition. So what does the person know? What are they no longer able to do that they could do before? Maybe they have trouble with language now, or maybe they have trouble with visual processing, or maybe they have trouble processing sound in general, or maybe they have trouble with memory. So looking at what part of the brain has changed and then seeing what that does to cognition. In more modern times, we can change the brain on a small time scale and reversibly, as we say. So changes to the brain need not be permanent. And now we can do experiments where someone might not be able to use part of their brain for a little while, but then it comes back. And we'll talk more about that toward the end today. But this is a kind of category of experiment where we change something about the brain and see what it does to someone's behavior, someone's cognition, and then we measure that. So we change the brain, measure the outcome psychologically. The alternative is the behavioral intervention approach. So we manipulate some cognitive process. Maybe the person learned something they didn't know before. Maybe they learned to navigate a maze or some other new space. Maybe we have them do math problems. We change something about what they're doing or what they know, some cognitive process, and then we measure the brain. So the idea here is that because of all of our behaviors, all of our thoughts depend on the brain, if we have a participant think about something or learn something, that's going to change the brain somehow, and then we can measure those neural variables, what the previous diagram calls somatic variables. Here we care about the brain, so specifically they're neural variables. So these are the two basic approaches. We can change the brain, see what it does to behavior, or we can change someone's behavior or influence their cognition, have them do something, and then see what that does to the brain. So when we talk about experiments, there are a number of categories that a given technique, a given research method will fall into. So a method might examine brain structure. So the parts of the brain. This could be large scale, being able to take images of the temporal lobe, occipital lobe, basal ganglia, brainstem. It could be very small scale, taking pictures of individual neurons or even parts of neurons. But all of these look at how the brain is put together. You can see the structure of neurons or the structure of groups of neurons in the brain, but you can't tell which ones are active. You can't tell what they're doing. You can't tell which parts of the brain are doing what. And so, all that said, this is still a very important approach. We have to know which neurons connect to which other neurons. We have to know what the parts of a neuron are. We have to know what the parts of the brain are, the parts of the nervous system. So some techniques will look at brain structure, while others will look at brain function. Is this neuron active or not? Is this part of the brain active or not? And then we can relate that back to what the person is doing at the time. If this part of the brain is really active, and the person is hopefully only doing one thing, it's an assumption, but often a valid one. If the person's doing something, and that part of the brain becomes active, and they stop doing it, and that part of the brain stops being active, it's a reasonable supposition that that part of the brain is responsible, at least in part, for that function. So a method can look at which neurons are active. It may not be very sensitive to what the structure of the brain is there, but they can tell whether those neurons, whichever ones they happen to be, whether they're active or not. And brain function, that is active neurons, can be detected in a number of ways. We can detect metabolic activity. This is a very common way of knowing which neurons are active. So here, the membrane potential is not measured. So we're not measuring action potentials. What we're measuring are things like oxygen usage, the usage of sugar, blood flow. So these are all necessary if a neuron is going to be active. It needs energy to do its information processing. So this shows up when we measure metabolic activity. This part of the brain is very active. Therefore, it needs more oxygen, it needs more sugar, it needs more blood. And so we can measure metabolic activity with techniques that can detect these variables. We can also measure electrical activity directly. So we can measure action potentials. We've already seen what an action potential looks like. And we're measuring those in terms of the movement of electrical charge. So we can do it that way too. We can also detect neural activity chemically. So if we have a way of measuring the concentration of a neurotransmitter, we can tell when a neuron releases that neurotransmitter. So we're not going to talk as much about that approach, but it is an important one, and it is used. Categorically, again, methods can either observe or manipulate. And this is an important distinction. So if I'm measuring something, I'm not changing anything about the brain. So I'm just observing. I'm recording data. So maybe it's structural data. Maybe it's functional data. So I'm measuring neural activity. That's one thing a technique can do, but it can also, instead, manipulate. It can change something about the brain. So instead of measuring the structure of the brain, a technique might manipulate it, might change the structure, might remove part of the brain or at least destroy it. It might change electrical activity in the brain. That would be a manipulation. Instead of measuring what the brain is doing, you're dictating what the brain is going to do. It can't use this part because you've shut it down or removed it. This part of the brain is going to be active now because I've stimulated it electrically, or I've shut it down electrically. So those are all manipulations of the brain, and those are different from observations, where I record what the brain sort of does naturally, whether the participant is engaged in a task or not. So I can either record data about what the brain is doing without interfering with the brain, or I can change the brain, I can manipulate it. Methods can also be what we call invasive or non-invasive. Invasive techniques are those that break the body envelope. You have to open up the skull, for example, to get to the brain. Or you have to implant an electrode inside the brain. Or you destroy part of the brain. So these are all techniques that require whoever's using the technique to using it to break the body envelope, to break the skin, to open up the skull perhaps. And that's in contrast to non-invasive techniques. Certain imaging techniques especially, the person doesn't have anything implanted in the body, nothing breaks the skin, and so those techniques are called non-invasive. Some techniques require a simple injection, for example. Technically those are invasive. Those occupy sort of a gray zone. It's not something as invasive as putting an electrode into the brain or creating a lesion in the brain or opening up the skull. A simple injection, especially if it's an injection in the periphery of the body, like the arm, that then affects the brain, those are sometimes called non-invasive. We're not going to make a big deal out of that distinction here. But there are a number of non-invasive techniques, and these have an advantage in that people that aren't undergoing a medical procedure already probably aren't going to volunteer for an invasive procedure, just for basic science research. If you're volunteering for an experiment, you're probably not going to volunteer for one that involves opening up the skull or pushing an electrode into the brain. But you very well may volunteer for one that's non-invasive, that doesn't break the skin, that just measures brain activity in a way that doesn't involve opening up the skull, for example. So that's a distinction, invasive, non-invasive. So we'll start with imaging methods. And the first thing we have to ask ourselves is, what is an image, before we get to imaging methods? Well, an image is a picture, we know that. And we see here on the right two images. One is entirely grayscale, the other is grayscale and color images combined. The one on the left is a structural image. We see a brain lesion on the left part of this patient's brain. On the right, we have that grayscale structural image, but on top of it is a what we call pseudo-color functional image. So those red and yellow blobs represent neural activity. And when you look at functional images, this is the way they tend to be presented. You'll see a grayscale structural image, and then the color-coded functional image, because knowing where brain activity is occurring is critical. Knowing that neural activity is occurring somewhere isn't very helpful unless you know where it's happening. More on that later, but when we ask what an image is, we know that it's a pictorial representation, and it's also an observational method. We are measuring the brain. We are not changing anything about the brain. This technique did not induce that brain lesion on the left. This technique did not induce that neural activity on the right. It's simply occurring. But we can measure how the brain is built. We can measure which parts of the brain are active. So these are observational techniques. And if you zoom really closely into an image, and you've probably done this with just pictures on your computer, you'll start to see the pixels, the individual elements of that picture. Actually, they look like little squares if you zoom in far enough, which is why a low-resolution image looks grainy. You can see the squares, which are called pixels. But each pixel just represents a number. So in this image, you can see that the bone of the skull shows up very brightly. Well, that means that those numbers are larger numbers. So when this technique was measured, when this image was taken, bone shows up brightly because the measurement technique represents bone with high numbers. And it represents those with a brighter color. Right next to it, air or cerebrospinal fluid shows up as dark because this imaging technique records that physical quality as a low number. And so you can tell the difference between different parts of the head and the brain because each individual pixel encodes a number, a measurement. And you can use imaging methods with either somatic or behavioral interventions. You can change brain structure and see what happened to the brain structurally with an imaging technique. Or you can have the person do something and see what that does to neural activity, a behavioral intervention. So now some examples. And we'll start with computerized axial tomography, sometimes just called computerized tomography, CT. But you've probably heard of a CAT scan, so that's the acronym I'll use here. And you get images like this, black and white images showing the structure of the brain. You can see the internal structure. This is a non-invasive technique. So it does not involve breaking the skin. It doesn't involve injecting anything. It certainly doesn't involve opening up the skull. So the person can go into the CAT scan machine and you can take images of the brain. And this is effectively just a fancy X-ray. It happens to be a three-dimensional one, but it uses the same basic idea as a conventional X-ray. And what it does, we're not going to go into the math or the physics, but what this technique does is take that X-ray from many different angles. So this is a cartoon example where you're taking an image of this cylinder and this rectangular object, but from one perspective, if you take a 2D image, it will look a certain way. As you rotate around the object, the relative position of those objects changes. Their angle to you changes. And so your two-dimensional image will change as you rotate around whatever it is you're imaging. And obviously the brain and the skull are more complicated than the cartoon objects we see here, but the principle is the same. If you rotate around whatever it is you're imaging, while taking a continuous series of 2D images, you can put all those 2D images together and then reconstruct a 3D image. And that's what a CAT scan is. It's a 3D X-ray. But it looks at brain structure. It's an observation. It's not changing the structure of the brain. And it's non-invasive. It doesn't involve breaking the skin. Another structural imaging technique is microscopy, using a microscope. And you get images like this one. And obviously here, you're seeing things at a very small scale. So you don't use microscopy to look at the whole brain. You can use a CAT scan for that, but you also don't use a CAT scan for looking at individual dendrites. You can use microscopy for that, so it's a difference in scale. So here we see black and white images of individual parts of a neuron. This is from Munoz-Cuevas et al., looking at dendritic spines. You can use fluorescent techniques. We're not going to, again, go into the physics here, but you can make particular neurons glow. That way you can see them better, and you can take an image of their individual parts. But again, we're not changing the structure of the neuron. We're just taking a picture of its current structure. And you can go beyond that. It's not just taking a picture. This, again, is from Munoz-Cuevas et al. And you can count dendritic spines. see when they show up. You take an image one day, take another image 2 days later, and you can see how structure in the brain has changed. So it's not just about taking the picture and getting a qualitative view. You can get quantitative. You can take measurements. How big is this neuron? How many dendrites does it have? How many dendritic spines does it have? So you can take measurements even after you collect these structural images. There is magnetic resonance imaging, or MRI. You may have had an MRI before. So again, not going to go into the physics, but you can use this technique to take a three-dimensional image of the brain. This is also non-invasive. Microscopy is invasive. You have to take a cell, take a group of neurons out of the brain, or open up the skull so you can see the brain without having the skull in the way. But MRI is a non-invasive technique. It's still observational, but it is non-invasive. And the physics are not important. It looks a lot like a CT scan. This happens to be a pseudo-colored image. MRIs tend to be in black and white. But it takes the image using a different physical method. And even though the images are similar, it will highlight different parts of the brain, different kinds of tissue, for example. MRI tends to be slower as well. So a CAT scan, resolution isn't quite as good, but it's quick. And so MRI tends to be slower, but you get a better quality image. So a lot of emergency rooms will use a CAT scan because time is of the essence. MRIs use an MRI machine like the one we see here on the right, and these employ enormous magnetic fields, which is why you can't take any metallic objects into the room with you. You have to be very careful about that. But this technique uses magnetic fields to examine the structure of the brain. So this is a structural technique. It's observational. We're not changing the brain. And it's non-invasive. It doesn't require opening up the skull to see the brain. A relatively new technique is diffusion tensor imaging. And this is a new technique, and it's been pretty exciting to see. Why it's called diffusion tensor imaging is not important. But what you get are images like this. And again, you have a grayscale image that is a structural image. That's not the DTI part. The DTI component of this image is the multicolored sort of wire-looking diagram. So you can see these little tubes, these little wires radiating throughout the brain. This is not, even though it's colorful, it is not representative of neural activity. What we're seeing here are the paths of axons. Not really at the level of individual neurons. It doesn't have that resolution. But axons tend to travel together. They form sort of bands or even nerves. And so this is bundles of axons. And you can see where these bundles of axons go. So we can see them here traveling from the center of the brain, areas like the basal ganglia, out to the cortex. So we can trace the paths that axons take using this technique. So it is structural. You're seeing what two parts of the brain neurons are connecting. They're going from point A to point B. You can see where their axons go. You can't see whether they're active or not. So this is a structural technique. It's also non-invasive. And so it's a technique very similar to MRI. Doesn't involve opening up the skull. Doesn't look at neural activity. It looks at neural structure. And again, not the level of individual neurons. Its resolution is not that good. But what it's doing here is taking advantage of the fact that this technique can measure where water goes effectively. And so basically it kind of tags water molecules, waits a little bit, and then measures where they are now. So you can see which way water moved. And again, this is not to look at neural activity, but water, if it's just sitting in a beaker, for example, if a water molecule is in one spot at one moment in the middle of the beaker, if you measure it a little bit later, it could be anywhere, really. It can move in any direction, and it is equally likely to move in any direction. It has no reason to prefer one direction over another. Axons are tubes. They're little containers. Water doesn't get out of them very easily. It's like having a pipe. Water doesn't get out of the pipe, it flows in one very particular direction. So similarly with axons, a water molecule that starts off inside an axon can either go up the axon into the cell body or down the axon toward the axon terminal. And the fact that the water is contained and can move in only a couple of directions is what we're measuring here. So we're looking at water molecules that are inside an axon and then seeing where they go, because they're only going to go where that axon goes. And that allows us to trace the axons to measure the paths that neurons take through the brain. So it's a structural technique, but it lets us see which parts of the brain are connected to which others. Another imaging technique is functional magnetic resonance imaging, often just called fMRI. So as the name suggests, this is a functional technique. MRI, without the F, looks at structure. fMRI looks at function, that is, neural activity. So this is the same basic idea as magnetic resonance imaging. So it's also observational, it's also non-invasive, but instead we're looking at neural activity. And you may have seen pictures like these in the media. So these colored blobs, they're often shown sort of looking warm or hot. They're red and yellow and orange. But what these indicate is neural activity. So the person is engaged in some task that requires the use of these parts of the brain. Those parts of the brain become more active, and we can measure that activity using fMRI. And what fMRI is really doing is measuring blood flow. So when an area of the brain is active, it needs more blood because it's using more sugar, using more oxygen. So blood flows to that area. And so we can measure that blood flow using fMRI. So we're looking at metabolic activity with fMRI. But that's a good indicator of which parts of the brain are active, which parts of the brain are engaged in whatever the person is trying to do. So you can see an image that looks like this, sort of a 3D brain. You can see a 2D brain, but you see the same basic idea, structural image, and then the functional image on top of it. So that's fMRI. It uses the same machine as MRI, but instead of looking at structure, it looks at function, and it does it non-invasively. So how does this work at a more detailed level? Well, we know about neural activity, we know about action potentials. So action potentials are electrical. However, neural activity requires sugar, it requires oxygen, and that increases blood flow. And that's what we're going to be measuring with fMRI, is increases in blood flow. Now, the way this really works, and the reason that we use effectively a big magnet like we do for MRI, is that that blood flow has a magnetic signature. So the molecule in your blood that carries oxygen is called hemoglobin. You've probably heard of it. But hemoglobin has iron at its core. This is one of the reasons you need iron in your diet. So these little hemoglobin molecules bind to oxygen when they're near your lungs and oxygen diffuses into your blood, and then they travel throughout the body. But because they have iron at their core, these hemoglobin molecules kind of act like little magnets themselves. So when you put them in a magnetic field, you can tell where they're going, and you can tell whether they have oxygen bound to them or not. So when an area is really active, it takes the oxygen away from those hemoglobin molecules so it can be consumed. So now those hemoglobin molecules no longer have oxygen on them, and we can tell the difference between hemoglobin that has oxygen and hemoglobin that doesn't because of this magnetic signature, because of the iron in these hemoglobin molecules. What does this get us? Well, we can measure the signal in a particular area using fMRI, and if an area is a little bit active, you'll see a characteristic response. You'll see a bump up in the fMRI signal. If you have a lot of neural activity, well, that area is going to be more active. It's going to need more oxygen. It's going to need more sugar. It's going to need more blood. And so we can see an even bigger signal. So we can tell not only which parts of the brain are active, we can tell, relatively speaking, how active they are by measuring the size of this magnetic signature. So that's how fMRI. Another technique is functional as well. It's positron emission tomography, often just called PET. You may have heard of a PET scan. You may have had a PET scan. So again, the physics are not terribly important, but the reason that it's called positron emission tomography, positrons are subatomic particles. They happen to be antimatter. So antimatter is not just in science fiction. It exists. It is a real thing. When a positron, which is emitted by a radioactive atom, hits an electron, they produce gamma rays, and we can detect these gamma rays. So effectively, a PET scanner is a big gamma ray detector. And the way this works is that you inject the person, so PET is arguably invasive, you inject the person with radioactive sugar molecules. Parts of the brain that need those sugar molecules will draw the radioactive molecules to them. And then when those molecules are there, they will release these positrons, and we can detect where the sugar molecules are, because those are the areas that release these gamma rays. That's where the radioactive sugar molecules are. It must be where the brain is active. And so you can see this brain activity in these pseudo-color images. This happens to be from your book, and you can see that there's more activity in the left image, that is a normal brain, than in the brain that has Alzheimer's disease. So we can look at relative neural activity using PET. So it's arguably invasive. It requires an injection. It's observational. We're not dictating which parts of the brain are active. We're measuring which parts of the brain are active. We're observing. And so it is a functional technique as well. We're looking at brain activity. And when it comes to these imaging techniques that look at activity, whether it's fMRI or PET, when we see the results, especially when we see the processed results in the media or in a research article, that's the result of data analysis. Those are not raw images of brain activity at any given time. So you take an average, you add things together, and functional imaging usually shows you a subtraction, especially fMRI. So you see the brain with those pseudo-colored blobs on it, and it makes it look like only part of the brain is active. And that also feeds into the you only use 10% of your brain myth. We know that's wrong. The whole brain is active. The whole brain is exhibiting some level of activity. It's just a question of how much, where are the differences. And so functional imaging often relies on a subtraction. So you have some really complex stimulus maybe that the visual cortex in your brain has to spend a lot of energy processing. You can compare it to visual cortex activity when it's not doing much, when you have a very simple image. And what you do is you have either a lot of participants or you collect a lot of responses to complex and simple stimuli, or both, and you average them all, you add them all together. And then what you do is you compare brain activity, those images, when the brain was processing the complex image, you compare it to the average activity when the brain was processing the simple image, and then you end up with the difference. And that's what you see in those pseudo-color images, is the difference between the stimulus condition and some control condition. And that's what those colored blobs are telling you. That's how big the difference is. That's where the difference is occurring. So if you're looking at differences in the processing of visual stimuli, you'll see blobs in the visual cortex, occipital lobe. But just to keep that in mind, it's not, when you're seeing those images, it is not a reflection of raw activity. It's a reflection of what we call a contrast, a subtraction between two different parts of the experiment. Some other observational techniques. So this is not an imaging technique, but it's an important technique. It's called electrophysiology. And we've already actually seen this, even though we may not have known it. So when you use an electrode to measure the membrane potential, you are using electrophysiology. Now this is a microscope image, and what we can see in this image are a couple of things. We can see some neurons, some cell bodies. You can see this little triangle part right here. That is the electrode. It's very small, especially the end of it that has to pierce the neuron. So it goes inside the neuron. So it's an invasive technique, not just because it's piercing the neuron, but because you have to open up the skull and pierce the brain to get to the neuron. But that's the electrode. It goes inside the neuron, and it measures the difference in charge between inside and outside the neuron. So again, we've already seen this. We did our unit on the action potential and looked at electrical activity. And we may not have thought about that as a research method at the time, but it is. It is looking at neural activity at the level of action potentials. So it's measuring electrical activity. It is an observational technique when you use it to measure an action potential. It's invasive, we already see that. And it is functional, and it's observational. So we can see an action potential. We are recording data. We are observing. We are not changing anything about the brain. So that's electrophysiology. And we can put a bunch of action potentials together. We can measure a neuron for a while, for example, and see that it has a response to a reward, or to a visual stimulus, or to an auditory stimulus. So you can measure electrophysiologically. You can look at neural activity. As I mentioned before, we're not going to spend a lot of time on this, but you can also measure chemical concentrations. You can say, oh, look, the neuron released dopamine. You can measure dopamine directly, not in terms of action potentials, but in terms of measuring whether a chemical is there or not. But electrophysiology looks at action potentials, looks at whether a neuron fired an action potential or not. And in this diagram in the lower left, you can see when the neuron fires action potentials. Each action potential is a black dot, and you can add them up over time and see what events the neuron is responding to using electrophysiology. On a larger scale, you can use what's called electroencephalography, or EEG. And this involves a cap on which a lot of electrodes are placed. So you have electrodes all over the surface of the skull, but it's non-invasive. You are not going into the skull. You are not breaking the skin. Normally, there are versions of this that are invasive. We're not going to worry about them. In general, EEG is done without breaking the skin. It puts electrodes on the surface of the scalp and then measures electrical activity, because the electrical signals that comprise action potentials, those electrical charges sort of diffuse. They're not just localized to neurons. You can detect them electrically by measuring electrical changes on the surface of the scalp. And it takes a whole bunch of electrodes. And this is a very fast technique. You can take thousands of measurements per second. Spatially, it's not very precise. So you'll have a series of these electrodes, but you don't have that many of them. And because you don't have that many, it's hard to localize where activity is coming from. So generally, with EEG, you may see the activity, but you may not know beyond an area the size of a silver dollar where that activity exactly is occurring. But what you get are what we call traces, time courses, of electrical activity. So this is an example here. We can see electrical activity across the various electrodes. One thing this is used for is examining epileptic seizures. So it doesn't treat epilepsy, but it measures when seizures occur. And because you get a rough idea of where electrical activity is occurring, you can get a rough idea of where the seizures are starting. But the electrical activity that occurs in the brain before a seizure is very different from what happens during the seizure. And so you get, during a seizure, these synchronized waves of electrical activity. So you can measure that, and that lets us know where the seizure is starting, lets us know the severity of the seizure in terms of electrical activity. And so this is used to help understand epilepsy in general, and also for a given patient to anticipate when seizures are going to start and to know where they're starting. So if surgery is necessary, you know where the surgery should occur. It's also used in sleep studies. So the different stages of sleep, and we don't get a chance to go into sleep in this class, they have different electrical patterns. They have different electrical signatures, and you can pick that up with EEG. You can put EEG electrodes on someone's scalp and tell what stage of sleep they're in. And obviously this is used not just to understand sleep in general, but if someone has trouble sleeping or doesn't have high quality sleep, you can see which stage of sleep they're in at any given moment and diagnose what the problem is. And at a basic science level, we can try to infer the function of different brain areas. So EEG is a fast technique. You can see very quick changes in brain activity. You can sort of tell where they're occurring. And it's really just good for measuring the outer cortex. You cannot see activity in the cerebellum, or basal ganglia, or brainstem, or even deep within the cortex. All you're really looking at is the surface of the brain. But if that surface is doing something, if it's involved in a particular task, you can see that, and you can infer that that part of the brain is involved in that task. So EEG is functional. You're looking at brain activity. It's non-invasive, just measuring the surface of the scalp. It doesn't break the skin. And it is observational. It doesn't change brain activity. It just observes it. And I'll go a little deeper on EEG because it's going to be important for our unit on attention. But EEG often uses what's called an event-related potential, or ERP. So you can measure individual responses to an event, to a stimulus. Not individual action potentials. EEG is averaging over large areas of the brain. But when you measure the response to a stimulus, it looks like a series of waves. And so for a given stimulus, as long as the stimulus is the same, and you're measuring the same part of the brain, that part of the brain will exhibit a characteristic response that we call an ERP, an event-related potential. We know that potential means a difference in voltage, and this is event-related. It is occurring after a particular stimulus, so it is related to that event. That's why it's called event-related, and again, we'll come back to this in our unit on attention when we see ERPs, event-related potentials, in response to stimuli that the person is trying to pay attention to. So a little sort of prelude there. So as I mentioned at the beginning, no one technique can do everything. There are always pros and cons, so we can compare our methods. So here we're going to compare functional methods, methods that look at neural activity. And one question you can ask is, how many neurons are we measuring? So electrophysiology measures just one neuron, or groups of neurons, small groups. You might measure maybe 100 neurons with electrophysiology. That's a very small number compared to the billions, the tens of billions that we have in the brain. So electrophysiology tends to be spatially very precise, but you don't get the whole picture. You only see a few neurons at a time, or maybe just one. In contrast, something like fMRI, or PET, or EEG measure large portions of the brain, or maybe the entire brain. You don't know what's happening in one neuron, or even in 10 neurons. You're averaging over billions, but you're getting the bigger picture. And so this is just a question of what it is you're trying to do. Are you trying to get a very good idea of what a particular neuron is sensitive to? Or are you trying to figure out where in the brain a particular process occurs? So again, there is no free lunch. No technique can do both. You either have to use something very spatially precise, but limited in terms of scope, or you have to use something that has a very broad scope that can measure the whole brain, but averages over billions of neurons. You'll miss out on the details. Another question you can ask is, how often does this get measured? How often are we measuring neural activity? So something like EEG measures very quickly. Electrophysiology measures very quickly. Very fine temporal resolution. Temporal means having to do with time. So we're measuring very often. Something like fMRI or PET, you might get an image every couple of seconds, which obviously you're going to not be able to see individual action potentials, because action potentials happen in 1,000th of a second. EEG and electrophysiology, you can see that. You can see individual action potentials and the results of them. Something like PET or fMRI, you're averaging not just over neurons, but also over time. So this diagram, this is not from your book, but we have a comparison of different techniques. So something can measure on the scale of milliseconds, or seconds, or even minutes. Spatially, something can measure individual neurons, or even parts of neurons, like a dendrite, or it can measure across the entire brain. So we have these pros and cons when it comes to spatial resolution and temporal resolution. And it's just a question of what it is you need to measure. Do you need fine temporal precision? Do you need to know exactly when this process starts? Well, then you might want to use electrophysiology or EEG. Do you need fine spatial resolution? Do you just really care about one neuron? Electrophysiology is good. Or do you need to know what's happening in the whole brain? Are you trying to figure out what process, what a given process does to the brain? Which parts of the brain are responsible for processing a particular kind of information? Then you want to look at EEG or fMRI or PET, especially if you want to look at deep structures in the brain. PET and fMRI are the only ones that can image the whole brain at once. So these are our two questions, our two ways of comparing methods. We've already seen observational versus manipulation. We've seen structure versus function. We've seen invasive versus non-invasive. And those are considerations. But we also have to think about how good is the technique for measuring whatever it is we're interested in. Okay, switching gears a little bit. Now we're going to change from looking at observations to looking at manipulations, somatic interventions. And one of the earliest ways of doing this was with lesion studies, either deliberately destroying part of the brain in animals or studying people who had had part of their brain destroyed by an accident or by disease. So this creates what we call a lesion, a damaged part of the brain that no longer works. And we call these lesion studies. The way these generally work, the sort of game plan for this, is that you measure behavior. So over here on the right, we can, for example, look at how a rat navigates a maze. So which arms of this maze it goes into looking for food. Then we induce some lesion. We destroy part of the brain. Then we look at how behavior changes. Before we destroyed part of the brain, maybe this rat knew where to find food. Then we destroy part of the brain, and now the rat sort of wanders aimlessly. So it either has lost the ability to navigate, or it doesn't remember where the food was. And so we can use this setup to figure out what a particular part of the brain, I'll call it brain area X, does. And it may take multiple experiments. Just from that one experiment, again, you may not know whether the rat has lost its general navigational skills or whether it's lost its memory for where things are. So you could do an additional experiment to test whether it really is memory or navigation in general. But that's how lesion studies work. We know what normal behavior is, we change the brain, we destroy part of it, and see what function is lost. Now there's some limitations, of course. Obviously in humans, we can't just go around inducing lesions in people's brains. It's not ethical. No one's going to volunteer for that either. But lesions do happen accidentally. One of the most famous patients in all of psychology is Phineas Gage, who we've already talked about. So Phineas Gage was in an accident and lost part of his frontal cortex. He was destroyed. And then people observed his behavior. And it's a little bit anecdotal because he wasn't actually part of an experiment. No one studied his behavior carefully before, but they could describe, people that knew him could describe what his behavior was like and then describe how he was different afterwards. In more modern times, lesions still happen accidentally, either due to injury or due to things like cancer, brain tumors, or due to surgery. One of the other most famous patients in psychology, patient HM, lost both hippocampi because he had surgery to remove them. Now, this was not done just for the sake of seeing what would happen. This was not an experiment. It was done to reduce his seizures. But as a side effect, his behavior was measured to see what losing that part of the brain did. And we know that he lost his memory, his ability to form new memories. Not to store them. His existing memories still existed. He could still retrieve them. More on that in our unit on learning and memory. But he could not form new ones. And from that, we knew that losing this part of the brain meant that you couldn't form new memories. So lesion studies are an older approach, but they can be very informative. Obviously, this is a structural change. It is a manipulation. It's obviously invasive, destroying part of the brain. And it is structural, technically speaking. Obviously, you lose brain function, and you can observe what functions have been lost. But what you're changing here is the structure of the brain. An interesting and relatively new technique is called transcranial magnetic stimulation, or just TMS. And what this does is, in one form, it stimulates the brain. If you do it over and over again, it actually shuts that part of the brain down. But this uses electromagnetism. So any electrical field will generate a magnetic field, and a magnetic field can induce electrical current. So there's a relationship between electricity and magnetism, and we're not going to go into the physics of it, but what TMS does is it uses a magnetic field to change electrical activity in the brain. And again, if this magnetic field is applied just once, you can cause action potentials to occur. If it's done repeatedly, you can sort of wear part of the brain out temporarily, so that part of the brain is no longer active. This is how it looks in real life, the real image. So you can stimulate that person's brain. If you stimulate motor cortex very quickly, you can make that person's finger move, for example. If you shut down part of the brain, then you can stop it from working temporarily. So now the person cannot use that part of the brain. If you do it over visual cortex, the person may not be able to process some aspects of a visual stimulus. If you do it over frontal cortex, it will change perhaps their decision making. So this is an somatic intervention. It is a manipulation, in other words. Those are the same thing. It is functional, though. We're not changing the brain's structure. Nothing about the brain will be different structurally after this. It's inducing activity or shutting down activity. So it is a functional technique. It's also non-invasive, and that's another advantage over a real lesion. This does not involve opening up the skin or opening up the skull or injecting anything. It just shuts part of the brain down for a little while. Now you have to be a little bit careful because if you're changing something of that person's behavior, you are responsible for them until their brain recovers. It takes about half an hour. But this is an interesting new technique because you can directly get at what part of the brain does. Imaging techniques are more correlational. You have them do something, you observe the brain, and you assume that the pattern of activity you see is due to what the task was. Here, you are directly changing the brain. So you know that the person can't do something that they could before, that it's because of the part of the brain that's been shut down. So that's TMS. Optogenetics, another interesting new technique. So here we see a rat that has undergone this optogenetic approach, and you'll see that there's this probe implanted in its skull and it's lit up. So what optogenetics does is it manipulates brain activity using light. That's a really interesting thing to do. The way this works, there are a couple different ways. You can either activate part of the brain, individual neurons even, using light, or you can shut them down. So for example, there are particular channels that don't exist normally in rats and mice, but if you genetically modify the rat or the mouse, it will do what we call express. It will have these special proteins, these special channels in the membranes of some of their neurons. Now, what that channel does is it opens up if you shine blue light on it, and it lets in sodium. And we know that letting in sodium will bring a neuron closer to threshold. It will make it more likely to fire. So that's one thing that optogenetics can do. You can genetically manipulate the rat or the mouse, and then you can turn a light on that's an electrode that's implanted in the brain, and induce neural activity. You can make part of the brain active and see what happens. You can also do the opposite. You can use a different protein, and if that protein is in the membrane of the neuron, it will, if you shine a yellow light on it, it will let in chloride. And chloride is negatively charged. If you let chloride into the neuron, it will inhibit the neuron. It will shut that neuron down. So this is a fascinating new approach, and it is new. So by manipulating the genetics of the rat or the mouse and having their neurons express these different channels, you can then selectively turn on or turn off particular neurons. And you can see what those neurons do. And again, this is very selective. Some neurons will express this and some neurons will not. So you're not shutting down the entire brain. You're shutting down very small numbers of neurons or activating very small numbers of neurons. So this is a powerful new technique. You obviously can't use it in humans because we can't genetically manipulate humans ethically or using this approach. But it lets us see what different parts of the brain do in the rat or the mouse. It lets us see the effects of changes in activity. And changes in activity at very particular times we can control with a great deal of temporal precision when the neurons are active or shut down. So new technique, very exciting. We can also stimulate the brain in animals. And this is an older technique, but it basically uses an implanted electrode, not unlike optogenetics. This breaks the skin, and you have to go through the skull. Optogenetics and electrical stimulation in animals are both invasive. They are both functional. We're not really changing brain structure, we're changing activity. And so this is a manipulation. So we're changing the brain. So electrical stimulation is something that's been going on for decades. So you can put a rat in a setup like this one. It has an electrode implanted in its brain. If it is implanted in the reward center of the brain, then the rat can push a lever, stimulate itself, and you can observe what the rat does. Turns out in this case, the rat will press that bar over and over again to stimulate its own reward center. It's not actually getting food or water, but that stimulation acts like a reward. And we've seen electrical stimulation as part of our unit on action potentials. You can induce an action potential in a neuron by stimulating it electrically. You can change what it's doing. So it's a functional technique, and it's a manipulation. There was a famous experiment decades ago by a scientist named Jose Delgado. And so what we see here is a picture of this experiment. What Delgado did was he implanted an electrode into the brain of a bull. And then he had a remote control that would activate the electrode. And so we'll see Delgado here describing this experiment.
Well, next day, the bull was normal, and the bull was just going around, charging against anybody who could be in the bull ring. It's not completely normal. Then I thought, well, I know a little about bullfighting, so I'm going to test this by myself. The animal was a few feet away. I push a button in the radio stimulator, and the animal really stopped completely. It was frightening, but it was an experiment.
Okay, so we see Delgado's experiment where he can change the bull's behavior. It stops charging when he stimulates that electrode. There were other versions of this experiment that he did, and one, for example, the neuron that he could stimulate made the bull moo every time he pressed it, every single time. Other behaviors as well. And this is not a particularly controlled experiment. It's happening in a bull ring, so and it's obviously invasive, and it's interesting. It was interesting at the time. Now it's sort of basic knowledge, so it's probably the experiment that wouldn't be done today. But it shows the powerful effect of electrical stimulation, that it changes behavior. And electrical stimulation is not just used for experiments, for basic science. It's also used for treatment. We've already seen in our unit on the brain how electrical stimulation is used to map the brain when a surgeon is trying to determine which areas to avoid when doing surgery for epilepsy, for example. Stimulating one part of the brain may cause a limb to move. Another part causes a sensation that the limb is being touched. Another location, that there's an odor. Another location, there's a visual stimulus. So by stimulating various parts of the brain electrically, the surgeon can tell which parts to avoid. Now, obviously, this is invasive. You're touching the brain, you're opening up the skull. It is a manipulation, and it is functional. You're not changing brain structure, you're changing brain activity. The same is true of deep brain stimulation, or DBS. In this case, though, this is not used to map the brain. This is used to stimulate the brain over and over again. So this is an implanted electrode. It is permanent. And what it does is it increases the activity level wherever the electrode is. So this happens to be in the basal ganglia, part of the brain responsible for reward processing and movement. For patients with Parkinson's, this does not cure Parkinson's, but it does alleviate some of the symptoms. Parkinson's occurs when dopamine neurons in the basal ganglia are dying. They're not exhibiting enough activity anymore because the neurons are dying. There are no longer enough neurons to exhibit the usual level of activity. So the electrode in DBS supplements activity in the basal ganglia, sort of takes over. Now it's not on constantly, but it does consist of sporadic bursts of electrical activity. So you're stimulating the brain over and over again periodically, and that alleviates some of the symptoms of Parkinson's. Recently, it's also been used for obsessive compulsive disorder, and we don't know exactly why. We don't know the exact cause of OCD, but stimulating the basal ganglia this way seems to help with some of the symptoms. Okay, that will do it for our unit on research methods. So we've seen different ways of conducting an experiment. We have things like functional techniques versus structural. We can be invasive or non-invasive. We can observe and we can manipulate. So different ways of describing experiments, different approaches, but they're all useful. And of course, we have that distinction between a behavioral and a somatic intervention. So we can either have the person do a task, or we can have them learn something and then see what happens to the brain, or we can change the brain's activity or its structure and see what happens to behavior. And imaging is a good example of these techniques. We have structural imaging. We also have functional imaging. So imaging is observational, but it can examine either structure or function. And that's a big category of our research methods. And then we also have somatic interventions that can excite neurons or inhibit them. Obviously, we can get rid of them entirely with lesions, but we can excite or inhibit them with TMS. We can do it with optogenetics. We can do it with electrical stimulation. So we have different ways of intervening in the brain, of manipulating the brain as well. And all of these can tell us what a brain area does, or how a behavior changes the brain, or how a disease changes the brain. Okay, so for next time, obviously be sure to take the online quiz for this lecture. And then for next time, read Chapter 4. So we're going to start our unit on psychoactive drugs. Also, coming up in the near future, instead of a research article for our upcoming exam, it will be a virtual lab experiment. So this is the link. I will send this out via email as well, but that is coming up on the horizon. So instead of reading a paper, you will participate in a virtual lab exercise. So I will see you next time.